Hello, Namaskar. Today I will be dealing on the topic type study, Marchantia, genus morphology and structure. I am Dr. Jayalakshmi, Assistant Professor of Botany, Postgraduate Department of Botany, Mahani Science College, Pullman, Mysore. So this is a topic of first sem BSc Botany under the syllabus of University of Mysore. Coming to the objectives dealt in this presentation is the study of the general features, that is occurrence, that is habit and habitat of Marchantia, its morphology and its anatomy. Let's start with the systematic position, that is it belongs to kingdom Plantae, division Bryophyta, class Hepatic Oxida, order Marchantialis, family Marchantiaceae, and genus is Marchantia. Coming to its occurrence, the genus is about there are 65 species all over the world. Preferably grows in best in cool, moist, and shady places. And Marchantia polymorpha grows as a pioneer in burnt forest side, or that is it's the first one which begins in the succession of the forest vegetation in the barren land. Seeing its distribution in India, there are 11 species which occur in India, mainly the western Himalayas. Marchantia polymorpha occurs at higher altitude in Himalayas on moist river banks and the rocks of the Himalayan belt. And Marchantia palmate occurs in Kashmir, Kuno, South India, Western Ghats. And Marchantia simlana occurs especially in Himachal Pradesh. And Marchantia nepalensis is very widely distributed in Punjab and Garhwal hills of Himalayas. Coming to its morphology, the vegetative structure, if we see, the Marchantia is a thalloid form. The plant body is the prostrate, dorsi-ventrally flattened thallus. So the dorsi-ventrally flattened thallus is dichotomously branched, and if we see the dorsal surface of the thallus, we can see that there is a prominent midrib. You can see the prominent midrib over here, which is darker in color, which deepens at the apex. At the apex, you can see the groove will become deep, and presence of gemma cups, which enclose a number of gemmae, is found along the midrib region of the thallus. And gemma is the asexual reproductive organ. So sex organs are born on special upright branches from the dorsal surface of the thallus. From the dorsal surface, we find erect structures which arises, that is antheridiophores or the gametophores, which are special upright branches. The gametophores which bear antheridia are called as antheridiophores. The gametophores which bear archegonia are called as archegoniophores. On the dorsal surface, we find polygonal areas with dot in the center, as you can see here, small pores in the center, which represents the air chambers, which is found in the internal structure. So we find polygonal areas on the dorsal surface of the thallus. Coming to the ventral surface of the thallus, it bears two appendages. One is the rhizoids and the scales, usually along the midrib. We find these structures associated usually along the midrib region. The rhizoids are unicellular and colorless. There are two types of rhizoids. One is smooth-walled rhizoids, where the inner walls are smooth. Another one is pegged rhizoids, where we find a peg-like or papilla-like projection within the internal inner wall of the rhizoids. The main function of these rhizoids is anchorage and absorption of water and minerals. They fix the thallus to the substratum and help in absorption of water and minerals from the substratum. The other appendages associated with the along the midrib region or the median scale, we find the scales. These scales are multicellular structure, that is usually multicellular structure with one cell in thickness, violet color due to the presence of anthocyanin pigments. They are arranged in two rows on either sides of the ventral groove. There are two types of the scales. One is appendiculate scales. These have an apical subrotate appendage, and from the inner row of the scales, the appendiculate scales are found in the inner row, while the other type is the ligulate scale. So you can see this is appendiculate scales with two lobes, while ligulate scale is just a finger-like projection. So these are small without appendage and form the outer rows of scales. We find the ligulate scales on the outer row and appendicular scales in the median or the midrib region. The main function of the scale is again protection of the growing point, retention of water by capillary action. So that is why the thallus can thrive even in the dry conditions, as water is retained within these scales and keeps the ventral surface moist. Coming to the internal structure or the anatomy of the thallus is differentiated to three regions. That is, when we take a section of the thallus, we can see three different regions: epidermal region, photosynthetic region, followed by that storage region. The epidermal region consists usually the upper and lower epidermis with a single layer of cells, which is interrupted on the dorsal surface, or the upper epidermis is interrupted by barrel-shaped air pores, which usually function for facilitation of exchange of gases. We find air pores on the in the upper epidermis, which mainly helps in exchange of gases. They are also called as chimney pores. Followed by that, we find the next region is a photosynthetic region, just which lies below the upper epidermis. So below that, we find the photosynthetic region, which is also called a assimilatory region. It consists of large air chambers. So this is the large air chamber, which is separated by single layered partition wall, and usually the cells are uniform in shape, arranged in a horizontal row. Many photosynthetic filaments or assimilatory filaments, which are rich in chloroplasts, are found arising from the base of this photosynthetic region, which is branched or unbranched, containing chloroplasts, which form the main assimilating portion of the thallus. Followed by that, we find the storage region, which lies below the photosynthetic region, which is a compact zone of several layers of polygonal parenchymatous cells, compactly arranged, and cells are devoid of chloroplasts, and there is no intercellular space in between. Each cell contains starch and protein granules. Mucilage is found in some cells, while storage region is thick in the center. So if we see the thallus section, the storage region is several cells thick in the center, and it is a few cells thickness as it or tapering towards the edge of the thallus, while several layers of thickness in the central portion of the thallus. The cells of the midrib region are elongated with reticulate sequences. So you can see in this region, the cells of the midrib are elongated, and followed by that you find the lower epidermis, which gives rise to the rhizoids and scales. So the thallus internally is differentiated into three regions: epidermal region, photosynthetic region, and storage region. So this is the anatomy of the Marchantia thallus. So these are the references which is being referred to prepare this presentation. Thank you.